Good morning, students. Today in this lecture talk, you're understanding about safety measures in microbiology laboratory. I'm discussing this as per first year, second Sem BSc Biotechnology NEP Practical syllabus. Okay, before going through NEP Practical, first we must understand safety measures in microbiology laboratory. Now let us go through the list of experiments under second semester Biotechnology NEP syllabus. First experiment to study the principle and applications of important instruments like biological safety cabinets, that is nothing but LAF, laminar airflow, autoclave, incubator, BOD incubator, hot air oven, light microscope, pH meter. These are the instruments you must study in this NEP microbiology syllabus, okay, which are used in microbiology and biotechnology laboratory instrument you are studying. Then experiment number 2, sterilization of media using autoclave and assessment for sterility. Third experiment, sterilization of glasswares using hot air oven and assessment for sterility. Fourth experiment, sterilization of heat-sensitive material by membrane filtration and assessment for sterility. Fifth experiment, preparation of culture media for bacteria, fungi, and their cultivation. Sixth experiment, plating techniques. In that you are studying and also performing spread plate, pour plate, and stick plate methods. Seventh experiment, isolation of bacteria and fungi from soil, water, and air. Eighth experiment, study of Rhizopus, Penicillium, Aspergillus using temporary mounds. Ninth experiment, colony characteristics study of bacteria from air exposure plate. 10th experiment, staining techniques like bacteria, example, gram staining, negative staining, capsule staining, endospore staining, and fungal stainings by using lactophenol cotton blue. 11th experiment, water analysis, MPN test. 12th experiment, biochemical test like IMBIC, starch hydrolysis, catalase test, gelatin hydrolysis test. 13th experiment, bacterial cell motility like hanging drop technique. So these are the various experiments you must perform in your second SEM BSc Biotechnology as per NEP syllabus. So before going through all these 13 different experiments, first and foremost important thing is that in microbiology laboratory, you must go through safety measures in microbiology laboratory. Now let us go through what are the safety measures you need to go through. After safety measures, even you must understand cleaning and sterilization of glassware. That will be dealt in other video. Okay, your learning objectives like here you're understanding in this semester you're going to understand in detail about microbiological tools and microbiological techniques. Okay, then here you are going to become more proficient at laboratory skills and safety measures, and also you are going to learn to follow experiential procedures and can also develop skills to formulate answerable questions or hypotheses, and also you can predict expected result while performing these experiments in microbiology. And further, if you want, you can also refer in microbiology by Pelzar, Microbiology Concepts and Applications by Paul, Fundamentals of Microbiology by Falbeizer and Saunders, Microbiology Ronald, Introductory Biotechnology RB Singh, then Industrial Microbiology, Casual, etc., Fundamentals of Bacteriology, Frontiers in Medical Technology, Biotechnology, International Trends of Perspectives, General Microbiology, CB Power. Also, you can refer College Microbiology, Volume 1 and Volume 2, written by Sundarajan. So these are some of the reference book you can purchase, and you can go through these particular books for you according to your syllabus. Okay, and further, whenever you enter your microbiology laboratory, compulsory to keep your books and your belongings in the rack. And also before doing any kind of experiment, first you need to wipe your working bench area by using surface sterilizing agent, and you need to keep all your place clean and thoroughly maintained condition. Okay, you need should not supposed to keep here and there in microbiology laboratory. That should be compulsory. Go the things whatever you are taken, no, that should go back to the places. and and please make sure in microbiology laboratory to keep first aid kits. Here you could see some of the instruments like pH meter, centrifuge, then here exposing the inoculation loop to the flame, incineration, and here you could see the streaking and the weighing balance, centrifuge, BOD incubator, and laminar airflow. Okay, then orbital shaker. Okay, then here flame, refrigerator, light microscope. Okay, here you can see the again orbital shaker, then weighing balance, hot air oven, laminar airflow, autoclave, etc. Okay, here you could see some of the glasswares like beaker, conical flask, test tube, micropipettes, glass pipettes, and petri plates. Here you can see various size of petri plates. You can use that in the uh, microbiology laboratory. Now let us understand here safety measures in microbiology laboratory. First and foremost important measure is that always wear a clean lab coat or apron before entering a lab for protecting clothes for avoiding contamination or accidental discoloration by staining solutions. Observe all possible cleanliness and neatness in case of apparatus, microscopes, and desk working surface area too. Do not use or wear lab coat while eating or drinking. Cut nails regularly. Tie long hair back to avoid contamination and fire hazard. Learn to keep paper, pencil, finger, and other objectives out of your mouth. Clean your working bench with 70% ethanol, that is surface sterilizing agent. Do not put your finger in your eyes, ear, and mouth. It may facilitate the chance of infection by pathogenic microorganisms. Okay, whatever instruction I have given here, kindly follow these instructions in your laboratory while working. Okay. Further, do not eat, drink, or talk while working with microorganisms. The inoculation needle is used in transferring the culture and should be sterilized in the flame before and after use. Flame the wire vertically only. Do not mishandle the chemicals, staining solutions, spirit lamp, UV light, instruments, or any other apparatus. In case the living cultures spill on the working bench, it should be covered with an efficient disinfectant and the hand should be washed thoroughly with soap and water. Never discard a living culture in the sink directly without sterilizing in an autoclave. Okay, because there may be chances of growing the same organism in the sink and they may contaminate. So always you need to kill the organisms by using autoclave. Then that can be thrown into the bin. Broth cultures must never be pipetted with mouth. Always keep the culture tubes in an upright position in the rack and maintain aseptic conditions while working. Do not open culture tubes or plates directly and never inhale them. Okay. Further, in case of personal accidents like cutting or pricking the finger or splashing of culture material in the eye, report immediately to the teacher in charge. It is must. Always clean the microscope stage, eyepiece, objective, and lens before and after use. Keep the lab doors and windows open as an experiment is in progress. Before leaving the lab, see that the gas and water are turned off. Even check the light also. Always wash your hands with soap and water before and after the close of an experiment. Keep a complete record of each day's work and keep the lab notebook up to date. After completion of the work, label the cultures with the name, code, and date of work, as it will help recording the data, and also that makes you to be in touch with the whatever the observation. Okay, so these are some of the safety measures in the microbiology laboratory. Thank you everyone for watching this video.